Now that you're familiar with the structure of a typical cell that makes up the human body, we can talk in more detail about each of the major components of those cells, beginning with the nucleus and working our way through the cytoplasm to the plasma membrane, the outer boundary of the cell. One of the things you should have noticed from the figure of the composite cell is that the nucleus is the largest organelle in the cell, and it's separated from the rest of the cell by a membrane called the nuclear envelope. Again, if you look at the figure of the composite cell, you can see the nucleus there in the center. At this point, however, it's difficult for you to see the fine details of the nucleus, so if we blow the figure up, we can take a closer look at the structure of the nucleus. First, notice that the nuclear envelope has two distinct layers. And that double layer of protection is very important because the nucleus contains some important structures, which must be protected and separated from the cytoplasm. So again, the outer boundary of the nucleus is composed of a double-layered nuclear envelope. And notice that the nuclear envelope has numerous holes or pores. The pores in the nuclear envelope are important because they allow certain molecules to move into and out of the nucleus, allowing the nucleus to communicate with the rest of the cell. For example, one of the things that moves from the nucleus into the cytoplasm is messenger RNA, which is important in the process of protein synthesis. Okay, you'll learn more about messenger RNA later on in the semester, but for now remember that the nucleus consists of this double-layered nuclear envelope which has numerous pores that allow for the exchange of molecules and information between the nucleus and the rest of the cell. Remember, we define the nucleus as the control center of the cell, and one of the processes it controls is cell reproduction. The cells that make up your body have a finite lifespan, so in other words, they don't live forever. But before those cells die, we want to make sure that they've been replaced. So the nucleus is important in regulating this process of cell reproduction. Okay, the nucleus is also important because within it is DNA. Okay, DNA is your genetic material. Okay, notice in the figure that you can see when inside the nucleus there are several dark purple strings, and those dark purple strings are labeled chromatin. Okay, chromatin is one form of DNA, okay, and when your cells are not dividing, the DNA in your cells exists in the form of chromatin, which is a thin, thread-like form of DNA. When your cells start to divide, however, in the processes of mitosis and meiosis, the chromatin will begin to condense with the help of some proteins and form a dark staining structure called a chromosome. Chromosomes are important because located along them are functional units called genes. Okay, genes contain the information needed to make specific proteins. And the function of genes and the process of protein synthesis is something you'll explore later in the semester. You should have noticed in looking at the figure of the nucleus the dark blob in the center labeled the nucleolus. The nucleolus is important because it's the site of ribosome synthesis. Okay, ribosomes are one of the many important cytoplasmic organelles found in a typical human cell. We'll look more closely at ribosomes shortly, but for now keep in mind that they are manufactured by the nucleolus, which is within the nucleus. Remember when we defined a cell, we said that cell biologists note that all cells have three main parts: the nucleus, cytoplasm, and a plasma membrane. But this is biology, and in biology there's usually an exception to every rule, as is the case here. Okay, most cells, like the macrophage you see in the figure, which is a type of white blood cell, have one nucleus. Okay, there are, however, some cells that have more than one nucleus. For example, the skeletal muscle cell shown here has more than one nucleus. Cells with multiple nuclei are called multinucleated cells. Other examples of multinucleated cells include bone-destroying cells and some liver cells. There's also an exception to the rule that says that all cells have a nucleus. Okay, this isn't entirely true because red blood cells, also called erythrocytes, are anucleate. Okay, this means that they do not have a nucleus. Take a look at the figure of the red blood cells. Okay, obviously, we know that red blood cells are important. Okay, these cells are responsible for transporting oxygen to all of the tissues in your body. Okay, remember before we said that the nucleus is the control center of the cell, and one of the things it controls or regulates is cell reproduction. So if a cell like these red blood cells have no nucleus, how do they reproduce? Okay, take a moment to think about this question before moving on. This is somewhat of a trick question, the reason being that red blood cells don't actually reproduce. Instead, red blood cells are produced by a special type of stem cell located in your red bone marrow. So red blood cells don't have a nucleus, they don't need a nucleus because they don't reproduce. Once a red blood cell reaches the end of its lifespan, it's simply broken down and replaced by new cells produced in the bone marrow. Moving outward from the nucleus, let's take a closer look at the cytoplasm. Cytoplasm is all of the cellular material between the nucleus and the outer plasma membrane. Cytoplasm is extremely important because it's in the cytoplasm that most cellular activities take place. Now, the cytoplasm itself can be divided into two main components. The fluid portion of the cytoplasm is called the cytosol, and scattered throughout the cytosol are various cytoplasmic organelles. Because organelles are responsible for carrying out all of the biochemical processes that keep a cell living, they are collectively referred to as the metabolic machinery of the cell. Okay, so once again, looking at the composite cell, you can see that the cytoplasm in the figure is represented in yellow. And notice that in this figure there's quite a bit going on. Okay, there's lots of stuff inside the cell. One thing I want to mention at this point is that although this composite figure shows you every possible organelle you might find in a cell, that doesn't necessarily mean that all of the cells in your body contain all of the organelles pictured here. Okay, remember there are over 200 different types of cells in the body, and we said that one of the things that makes those cells different is that they have different internal compositions, which means that those cells have different combinations of the organelles shown in the composite figure. Okay, so for example, some cells may have lots of smooth endoplasmic reticulum, whereas other cells may have none. So don't make the mistake of assuming that just because an organelle is pictured in the composite figure, that all of the cells in your body have all of the organelles that you see there. Now let's move on and talk about each of the organelles that you find in a cell. The first on your list is the ribosome. Okay, ribosomes are small spherical structures, and they're composed of RNA and protein. You find ribosomes in two main locations in the cell. First, you find them attached to rough endoplasmic reticulum, and you also find them freely floating throughout the cytoplasm. Okay, now, it may be difficult for you to see the ribosomes in the figure, and it may be even more difficult for you to see them in the figure in your notes. But hopefully you can see that ribosomes compared to other organelles are extremely small, and they look like little dots in the cytoplasm. Despite their size, ribosomes are extremely important because their main function is to produce proteins. So ribosomes are protein factories. Now, the structure of ribosomes and the process of protein production is more complicated than what I care to go into at this point. But for now, keep in mind that ribosomes are small spherical structures that are responsible for making proteins. The next organelle on the list is the endoplasmic reticulum. As you can see in looking at the composite figure, there are two types of endoplasmic reticulum: the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Together, the two types of endoplasmic reticulum form a network of tubes that extends throughout the cytoplasm. The two types have different structures and different functions, and so we'll talk about each separately, beginning with the rough endoplasmic reticulum. Okay, looking at the larger version of the rough endoplasmic reticulum, first take note of the overall shape of this organelle. Notice that it kind of looks like a stack of tubes that have been smashed together and attached to the nuclear envelope. Again, to highlight, here is the rough endoplasmic reticulum. One of the defining characteristics of the rough endoplasmic reticulum is the presence of ribosomes on its outer surface. If you were to look at the rough endoplasmic reticulum under a very powerful microscope, it would have a bumpy or studded appearance because of those ribosomes attached to it, hence the name rough ER. The rough endoplasmic reticulum has several important functions. First, the outer surface of this organelle is responsible for synthesizing phospholipids and cholesterol, both of which are important components of plasma membranes, which we'll take a closer look at in Part 3 of this week's lab. In addition to producing phospholipids and cholesterol, probably the most important function of the rough ER is its role in storing protein products and transporting them to other parts of the cell. Remember, there are ribosomes attached to the outer surface of the rough ER, and that ribosomes make proteins. So the question is, where do those proteins go when they're made? In order to better understand what happens, take a look at the next figure. Ribosomes that are attached to the outer surface of the rough endoplasmic reticulum make a protein, and as that protein is produced, it enters the space of the rough endoplasmic reticulum. In this figure, the protein that's being produced is the purple beads on a string structure that you see there, and it's labeled a polypeptide. A polypeptide is another name for a protein. Once inside the space of the rough endoplasmic reticulum, a polypeptide or protein chain can be modified somehow. In this particular figure, we're modifying the protein by attaching sugar groups to it, and those sugars are attached to a molecule forming a glycoprotein. Once the glycoprotein is produced, the membrane of the rough endoplasmic reticulum will fold in around it and eventually pinch off, forming a vesicle that contains the glycoprotein. After the vesicle containing the glycoprotein has pinched off from the rough ER, it's shipped on to another part of the cell. So again, this process here is very important. Ribosomes attached to the rough ER make a protein. The protein enters the rough ER where it's modified, and once it's modified, the protein is packaged in a transport vesicle and shipped to another part of the cell. Now let's take a closer look at the smooth endoplasmic reticulum. Although the smooth ER is continuous with the rough ER, it has a much different shape and a much different function. So take a moment and look at the structure of the smooth ER and compare it with that of the rough ER. Whereas the rough endoplasmic reticulum looks like a stack of flattened tubes, the smooth ER has more of a looped structure to it, and it kind of looks like sea coral. Also, one of the characteristics that distinguishes smooth ER from the rough ER is that smooth ER does not have ribosomes attached to it. The smooth endoplasmic reticulum, like the rough endoplasmic reticulum, has several functions. The smooth ER, for example, is a site of steroid and lipid synthesis, lipid metabolism, and most importantly, drug detoxification. Because of its function, you should expect to find smooth ER in the cells of organs responsible for filtering the blood and removing toxins like alcohol, for example. So, for example, cells of the liver will be very rich in smooth endoplasmic reticulum. Moving on, the next organelle on the list is the Golgi apparatus. Take a moment to look at the Golgi apparatus in the composite figure. Notice that in comparing the overall structure of the Golgi with that of the rough endoplasmic reticulum, they sort of have a similar appearance in that they both look like a stack of flattened tubes. One main difference, though, is that the Golgi apparatus has large swollen ends. And those large swollen ends are one of the defining characteristics of the Golgi apparatus. All right, let's take a closer look at the Golgi apparatus. Notice in this figure that the Golgi has two different sides. One side is called the receiving side, and the other side is called the shipping side. Now, the Golgi apparatus is one of the easier organelles to remember, not only because of its shape, but also because of its function. And that function is fairly easy to remember if you think of the Golgi as being the UPS or the FedEx of the cell. So for example, let's assume that you buy a gift for a family member who lives in California, and that gift is breakable. And you decide the best way to get the gift to your family member is to ship it using UPS. So you go over to the UPS store, and the first thing they do with your gift is take it and wrap it in bubble wrap so that it doesn't get broken. They put it down in a box, tape the box up, and then finally ship it on to your relative. So the same basic process is happening here at the Golgi apparatus. It receives some sort of package, it processes the package and modifies it, and then repackages it and ships it on to some other part of the cell. Notice that in this particular figure there is a transport vesicle from the rough endoplasmic reticulum approaching the receiving side of the Golgi. Think back to the process we discussed with the rough endoplasmic reticulum and the ribosomes attached to it producing proteins. Remember, those ribosomes make a protein, the protein enters the rough ER where it's modified to form a glycoprotein, and the glycoprotein is then packaged and shipped to another part of the cell. The place in the cell where that glycoprotein is shipped is here to the Golgi apparatus. So once the transport vesicle reaches the Golgi apparatus, the membranes fuse and the glycoprotein is released into the Golgi. Inside the Golgi, the glycoprotein will be further modified. It may tack on a few additional sugar groups. Some of the sugar groups that were added in the rough ER may be removed, so that by the time the glycoprotein passes through the Golgi apparatus and reaches the other side, it's ready to be repackaged in another vesicle that will carry the glycoprotein to the plasma membrane, where it will be either incorporated into the plasma membrane or released into the extracellular space surrounding the cell. So again, the Golgi apparatus is very important. Think of it as being the UPS or the FedEx of the cell. It receives packages, it processes those packages, and ships them to some final destination. The next two organelles on the list are lysosomes and peroxisomes, and because they're very similar, we'll discuss them together. So let's first highlight a lysosome and then a peroxisome. Both lysosomes and peroxisomes are membranous vesicles that contain enzymes, so structurally they're very similar. The main difference between the two organelles is the type of enzymes they contain. For example, peroxisomes contain oxidase enzymes responsible for breaking down harmful oxygen-containing compounds like alcohol and hydrogen peroxide, which is produced as a byproduct of metabolic reactions that occur in the body. Lysosomes, on the other hand, contain acid hydrolases, enzymes that act as cellular digesters, breaking down foreign particles that enter a cell and old cell parts that maybe aren't working like they're supposed to. The enzymes that are found in both lysosomes and peroxisomes must first pass through the Golgi apparatus before they become active. If these enzymes bypass the Golgi, they would not be functional. So in this way, you can see that the Golgi apparatus, in addition to its function as the UPS of the cell, is important for the proper functioning of lysosomes and peroxisomes. Because the function of lysosomes is so important, let's take a closer look at its structure and function. First, take a look at the left side of the figure. Notice here that there's a small green particle labeled food. Now, this green food particle can be a number of things. It can be a bacterium trying to enter the cell. It can be a virus, something foreign to the cell that could potentially cause harm to the cell and to the body as a whole. In this example, let's assume that the green food particle is some sort of harmful bacteria. As the bacteria enters the cell, it's encased in a vesicle that's composed of pieces of the cell's plasma membrane. So once the bacteria enters the cell, it's enclosed in this food vacuole. Okay, the cell then recognizes that there's a foreign substance in the cell, something that shouldn't be there. And when the cell sees the food vacuole containing that bacteria, lysosomes containing active hydrolase enzymes are released, and the lysosomes then bind to the food vacuole and release the enzymes they contain into it. The digestive enzymes then begin to break down the bacteria, preventing it from replicating and doing further damage to the cell. Notice again that this process begins at the rough endoplasmic reticulum. The enzymes are produced in an inactive form, passed along to the Golgi where they are modified and activated, and then repackaged into a lysosome. Without this process, lysosomes would be incapable of breaking down bacteria that entered the cell. The right side of the figure shows the second main function of the lysosome, and that is digesting old cell parts. In this case, we see a mitochondrion that's been engulfed by a lysosome. Okay, mitochondria are responsible for producing cellular energy. So in this particular case, the mitochondrion may not be working like it used to. It may not be producing as much energy as it should. So the lysosome engulfs it, allowing its enzymes to break it down and recycle its pieces. So again, remember the main function of lysosomes is to function as cellular digesters. The next figure here shows active lysosomes in a cell. As you can see, the bubbles inside the lysosome represent areas of active digestion. The next organelle on the list is the mitochondrion, which we can see here in the composite cell. If we take a closer look at the mitochondrion, notice that overall it has sort of a bean shape to it. And it's composed of two separate membranes, the outer membrane and the inner membrane. Okay, mitochondria are nicknamed the powerhouses of the cell because it's across the wall of the inner membrane that cellular energy in the form of ATP is produced. ATP produced by mitochondria is used to drive many chemical processes that take place in the body. So for that reason, mitochondria are vital to the proper functioning of a cell and thus to the entire human body. Okay, one additional interesting note about mitochondria is that they have their own DNA, which means that they, unlike other cellular organelles, can reproduce themselves. The next organelle on your list are the centrioles. Centrioles are paired cylindrical organelles that lie at right angles to each other next to the nucleus. Because the centrioles are a little difficult to see in this figure, we can blow them up and take a closer look. So now you can get a better idea of what these things look like. During cell reproduction, the centrioles form something called the mitotic spindle, which is necessary to partition DNA into each of the new cells that's produced as a result of cell division. Okay, you'll learn more about centrioles and their role in cell division when you study mitosis and the cell cycle. Okay, one additional function of centrioles that I will mention at this point is that these organelles also form the basis of cilia and flagella, which are cellular extensions that allow the cell to move. The last three organelles on the list, the microtubules, microfilaments, and intermediate filaments, you can see here in the figure. We're not going to go into much detail with these three organelles, but basically what I want you to know about them is that they form something called the cytoskeleton. The cytoskeleton is important in maintaining the overall shape of a cell and in helping the cell resist mechanical stresses that would normally cause a cell to rupture or break open. That concludes Part 2 of Lab 2, the cell. Before moving on to Part 3, review the structure of the composite cell and each of the organelles shown in the figure in your notes.